organization dedicated to transforming the riverfront. For nearly 15 years, Riverscape has focused on the beautification and redevelopment of the Wabash River corridor in Vigo County. Riverscape is pleased to bring waterways to West Terre Haute, one of 6 small towns in Indiana selected to host the exhibit. Waterways is a Smithsonian institution traveling exhibition that reveals the central nature of water in our lives. It is made possible by the Museum on Main Street partnership between the Smithsonian and Indiana Humanities, with support from many local community partners and volunteers. Alongside the Smithsonian display, Riverscape hosts a companion exhibit, Pearls of the Wabash, which explores the local community's history and relationship to the Wabash River. The exhibit will be at the Vigo County School Corporation Administration Building at 501 West Olive Street, West Terre Haute through January 2nd, 2022. We are also excited to offer this Waterways Speaker Series, which taps regional experts on the local history, life, and health of the Wabash River watershed in partnership with the Vigo County Public Library. And our speaker this evening is Brant Fisher. Brant serves as the Nongame Aquatic Biologist for the Indiana Department of Natural Resources, Division of Fish and Wildlife, Office of Science and Research, Fisheries and Aquatic Sciences Unit. I work in higher ed. That's like a higher ed kind of title.
Yeah,
it's a lot of higher ed kind of title. Brant earned a bachelor's degree in biology from Grove City College in Pennsylvania and a master's degree in aquatic sciences from Purdue University. And he will speak to us this evening about conservation strategies for Indiana's freshwater mussels. Welcome, Brant.
Thank you. Hopefully you'll be clapping at the end too. I hope everyone can see the screen okay. It seems like it's doing pretty good. So you might be wondering what a non-game aquatic biologist does, since that's my title. So I thought I usually start with that. So non-game, so that means I work with species that you normally don't go out and fish for. Aquatic, so stuff in the water. So mainly non-game fish, which we have about 200 different species of fish in Indiana, which maybe 50 would be considered kind of sport or game fish, but then there's a whole other 150 species that I work with, mainly the endangered ones and listed ones. And then freshwater mussels is another big group, and then a little bit with snails, a little bit with crayfish, and a little bit with anything else that is in the water that people have a question about and don't have an answer. I don't have an answer for a lot of those other groups either, because I only got so much time to do stuff. But today, we're going to talk about the one big group that I spent a lot of time working with, that's freshwater mussels. And I'm going to talk about some specific projects we're doing. I'm not going to get a lot into, this isn't like a biology talk on what a mussel is and some of that stuff, but we'll delve into a little bit of that as we're going through this. But certainly if you have questions while we're going through the talk, feel free to interrupt me and ask a question. I may say, wait, we're going to get to it a few slides down the road, or we can just stop and talk about it, and that's fine. I can get off topic very easily, so if you want to talk about something, just ask a question anytime throughout this. So I always start with this slide, and this slide just kind of shows you kind of where the state is in terms of our mussel diversity and where we're at. So the 79 there that you see, that's how many historic species of freshwater mussels we have in the state or are known from the state. And some of that's from very old shell material, that species hasn't been alive for maybe 100 years in Indiana, but their shell material still remains. So when you go out and do surveys, you can find shell material stuff. And so we've historically documented 79 species of freshwater mussels. Currently, of those 79, we consider 17 of those to be extirpated, which means you can't find them live anywhere in Indiana anymore. And then another 11 are on our endangered list, which those species tend to be left maybe in one river or one section of one river. They're very rare in the state. And then our special concern species, we have 14 on our list right now. That's kind of just a step down from endangered. It's kind of ones that are doing okay, but we need to kind of keep an eye. They might be trending down. And so as you can see, of our 79 species that we know historically from the state, over half of them are either already gone or they're listed in some way. So for sure, freshwater mussels are definitely one of our most imperiled groups of animals we have in the state. And I always throw this slide up too because just to give you an idea, everyone thinks about federally listed species. Well, we actually have 19 federally listed endangered species of mussels in Indiana, and 5 that probably would be listed federally, but they're already extinct from the world. So, you know, they never made the list because they've been gone for a while. But the ones I have listed there, and you can see some of the names, those are the ones of those 19 that we actually still have live in the state. Quite a few of those ones that are federally endangered are on that extirpated list are already gone. I have kind of in parentheses there at the end, the rough pigtail white cat's paw. Technically, we do not consider those extirpated yet from the state, but there's probably a very good chance that they are. We have not seen a live one in the state for a very long time. And then we have 1 federal threatened species, and then we have 4 additional ones that are right now in some sort of the review process in terms of being listed or not. So we have a lot of federally listed mussel species in the state. For example, we have no federally listed fish species. Although there is one right now that is being petitioned to be listed, the lake sturgeon, and we'll kind of see how that goes down the road, whether it does get listed or not. So you might be thinking, wow, why is this group of animals so endangered in our state? And it's really hard to point to just one cause. So there's kind of a grouping of causes that probably all played a part in it. They live in the water, so certainly water quality issues over the years have probably played a big role. And a mussel is usually buried down the bottom of the stream or lake or pond. It's filtering water. So not only point source pollutions, but a lot of non-point source or sediments and stuff like that in the stream could certainly cause issues for some of our species. There's probably were a lot of catastrophic point source events that happened in our streams that wiped out mussels and stretches. And mussels have 1 foot that basically looks like your tongue. And that's the only way they can move as adults. So you can imagine they can't just get like a fish swim back up into a stream that's spent once water quality gets better. They need some help along the way to get there. And that's kind of some of the stuff we're going to talk about later. So water quality is definitely one of the reasons. Habitat, you know, there's a lot of streams in Indiana that occasionally get dredged or dipped out. For a mussel, that's not great because a mussel, once it's up in the spoil pile on the side of the, you know, the stream or ditch, you know, that's not a place that it can live. It needs to be in the water. It needs to have water. We certainly have some streams that even go dry now that maybe didn't go dry 100 years ago based on different reasons. Dams have played a large role in some of our mussels being more endangered and even to the point of extirpation. Some of our species really like the fast flowing riffle type sections of streams, and we put a dam and, you know, it pounds the water. So they don't do very well sometimes in that habitat. And then it also can block fish movements in some cases. And as we're going to see in a little bit, the fish play a very important role in the reproduction of mussels. And then, you know, overharvest is a really hard thing to get your brain around because there wasn't a lot of good data before harvest started to know if actually it was overharvest. But I mean, you can see in the right side picture, those are guys at a button factory sitting on a pile of shells. So I mean, there obviously was a lot of mussels taken out of our streams. Whether it was the button industry, then that went away like in the 40s when plastics came out. And then it was the cultured pearl industry where the parts of our shell were ground down and used as a nucleus for creating cultured pearls in some Asian countries. So that was a thing until 1991, and then the season was closed in 1991 in Indiana, and since then it is now illegal to harvest live mussels for any reason or even to take shell material. Some people don't know that, and I'm sure the law, you know, it wasn't put there to, you know, bust a kid picking up a shell, you know, to put in his pocket, but that is technically the law. It is illegal to take any shell material of our native species live or dead. And then non-native species have played a role as well. The bottom left, those are zebra mussels. Most people have heard of zebra mussels. Well, actually, probably when you hear the word mussel, you might actually think it's zebra mussels before you think of much else because they've gotten a lot of publicity. So what they do, as you can see there, they actually will grow on any hard substrate in a body of water. And so our mussels, they're buried in the bottom of the substrate, and a zebra mussel attaches to it, and then another one, another one, another one, another one. Eventually, it gets to a glob like that sometimes, and then that native mussel can no longer filter water, can't get food, can't get oxygen. So it can, you know, play a role in killing some of our mussels if zebra mussels are too thick in certain areas. Fortunately, they do not do very well in flowing waters. So a lot of our streams have mussels. Some of our rare species are in some flowing waters, and the zebra mussels, while they can get in there, they never get in very big numbers. So in Indiana, zebra mussels have had the biggest impact on some of our natural lakes in the northern part of the state and the Ohio River, because it acts like basically big impoundments. Zebra mussels have done very well in there. Yep.
Do zebra mussels actually filter the water like other mussels? Do they actually do any good for a waterway even though they're invasive?
That's a good question. They do exactly what our mussels do. They're filter feeders just like our mussels are. And when they get in an area, their populations can just blow up. So they can be filtering a complete lake, in days or hours sometimes, depending on how big the lake is. So what we've seen happen in some of our lakes that they've gotten into is they will actually clean the water because they're filtering it so much that they actually take a lot of the stuff out of the water. They make it clear, and then the sunlight can get deeper, and then plants can grow more. So sometimes when you get them in, it causes like a secondary effect where now you get more plant growth. Then that causes a whole nother sometimes issues in certain places. So yes, they are filter feeders. So whether they are cleaning the water in some instances, but to say that it's something you'd want to have, I think most people would probably still say no.
Were they introduced, or are they in the water by accident?
Yeah, so they came in from ships, like coming up through the Great Lakes, so ballast water. And then the Asian clam, they've been around in Indiana waterways for over 100 years. And you've probably seen them if you've been in about any river or stream, even some lakes. They don't get any bigger than about the size of a quarter. They're only yellow, this kind of yellowish color when they're live. When they die, they get much browner and darker. And they do not attach to mussels, but they just live in the substrate with our mussels, and sometimes lots. And so what their effects are is a little harder to understand. They could be competing for food. Usually in flowing waters, food isn't a complete big issue. But think about other things. Males are releasing sperm into the water, or native mussels for females to take in to fertilize eggs. If there's hundreds of thousands of these corbicula, Asian clam, all around our native mussels filtering the water constantly, they could be filtering all that stuff out, so it could interrupt some stuff for reproduction. I mean, there's a lot of other effects that the Asian clams could have. And then the bottom right is something that's just showed up in Indiana the last couple of years. That's a black carp. You've probably all seen silver carp, the Asian carp that jump out of the water and hit people in the head in the Wabash River, and grass carp, which have been put in a lot of places to control plant growth and stuff like that. And then there's also the big head carp, which is just kind of like a silver carp, it just doesn't jump. Well, the other one is the black carp. And this species is just a molluscivore. All it eats is snails and mussels. And it has just made its way into Indiana in the last couple years. In the Ohio River and the lower Wabash. I'm sure it's right here in Terre Haute in the Wabash by now. Yep.
Somehow I had the impression that mussels prefer moving water, but you're saying that they're in lakes too.
There are a handful of species that actually do very well in lakes, and actually they love lakes. The rare ones tend to be ones that are in our rivers and streams. And the ones we've lost are almost all ones that were in rivers and streams and needed that very good flowing water. But we have a lot of species that will do very well in lakes. In fact, I call them mud-loving species. They love mud. You always think of these pristine places, that's the only place the mussels we know. There's a lot of native species that do very well, kind of in different types of spots like that. Like you could probably go into a lot of the oxbow sloughs off the Wabash and you'll find mussels living in those places, and they're the ones that like that type of habitat.
Did you say that it's illegal to pick up mussel shell just lay there on the riverbank even if it's dead?
Yep, technically it is illegal to take or possess mussels live or just the shell material.
Is that also in Illinois?
It might be slightly different in Illinois. And in fact, in Illinois, I think it's still legal in some instances to take a live mussel for bait. And I think you can actually get a commercial license still in Illinois. But don't quote me on that. That's probably going on the video. But I look that up before you pick up a shell in Illinois to make sure. All right, I better speed up whenever you throw these. And another reason they're so endangered is just like people don't really know anything about mussels, so there's some apathy to them. They don't really care maybe as much about mussels. So things they could do to help mussels out, which is kind of hard anyways to think about, but really anything you can do to improve water quality or help water quality is going to be a positive for mussels. So we actually did a project up on the Tippecanoe River where we put some signage out. We did some kind of surveys. We did some education, some post surveys. We even had this person there up in the upper right. I think it looks more like a butterfly than a mussel, but it was supposed to be a mussel. It's hard to make a mussel costume, And we did some education, trying to educate people, especially recreational people. Like a lot of our rivers and streams that have our rarest mussels get a lot of recreation. So kind of trying to come up with things. What could people do to help out our mussels? So one was, pick up your canoe in the shallow spots and don't drag it across, and like maybe it's impact mussels in that way and some stuff like that. So Yep.
Before we get too far away from the black carp,
Okay.
do they eat more than the silver carp mussels?
For sure. Yes. They key in on wanting to eat snails and mussels. They're a molluscivore. The way so these fish have not teeth like you think of teeth. They got teeth in the back of their throat they use to crunch up stuff, and they got these really good crunchers that can actually crunch up shell material. And a silver carp wouldn't really eat a mussel ever.
It won't?
No, they're more filter feeders.
But I thought someone told us last week that the silver carp eats a lot of the same food as the mussels.
Okay, yes, because they're filter feeders. They're very similar to filtering the water like our native mussels will, but they wouldn't eat a mussel specifically.
So
what's the impact of silver carp?
what's the impact of black carp?
Yeah, it's going to be very interesting to see. Unfortunately, it's just going to be another hit on our mussels, from a brand new thing coming in. Yeah, it could be bad. We'll see. They're doing studies right now to kind of see, is our black carp just going after certain species, maybe thinner shelled species that are easier to crunch up compared to some of the thicker shelled species. So we'll see where that all plays out. But.
Now these mussels, when they start out, are they small?
Yeah, just like anything else, when they're like the size of a grain of sand when a mussel first falls off of a fish. And we're going to talk about that very soon here. And then, some mussels can get the size of a dinner plate when they're full-grown adults. But yeah, they're very small when they first start out.
Is the population coming back at all? Is it?
We're hoping. Yeah, I think in some instances we're seeing some rebounds for some species, but some species still continue to have kind of a downward trend. So that's what I'm going to talk about here. Some of the things we're actually doing to try to help out some of our population. That's what I'm going to focus on now, a couple of our projects. So here's kind of a list of some of the different types of restoration we might do with mussels. And I'm going to hit on each one of those a little bit. The first few, though, I have some very specific projects that we're involved in right now. So one is propagation. Just like we have fish hatcheries, we raise fish. We can do things where we can actually raise mussels to put back places. So the one type of propagation we do right now is a cage culture type. And we're doing that with a federally endangered species. It's called the snuff box. And I don't know if it gets its name from kind of its shape and what it looks like. They used to use shells actually outside of actual snuff boxes that people used and they used to make. But that's the species we're working with now. It's actually one of our species of mussels where it's sexually dimorphic. So you can actually, when you pick up a snuff box, you can tell if it's a male or a female based on the shape of the shell and some other characteristics. The two on the left are what a male look like. The two on the right are what a female look like. And the female has these serrated edges to its shell and has kind of more an extended part of its shell. Usually a female, a lot of times, will look a little different than the male because they're holding eggs in their gills. So they need a little different space in there than a male would need. So that's nice for this project because when we're looking for females to use for the propagation work, which we have to get our hands on, we can actually tell them apart very easily just by as soon as we pick them up. So this is kind of the distribution that we know of for snuffbox in the state of Indiana. Now the red and orange dots are spots in the last 30 years we've found live individuals or what we call fresh dead, meaning it's both halves of the valve open and it obviously has died very recently. All the other lighter colored dots are places just where we found old shell material. So you can see its distribution in the state was pretty decent. It was in the Wabash right here through Terre Haute at one point in time. But really, this population down there, that's the upper East Fork White River drainage. That's like Sugar Creek area, if you're familiar with that. Not the Sugar Creek that, we have too many Sugar Creeks in Indiana. Not the Sugar Creek that goes through Turkey Run into the Wabash, but this is the Sugar Creek that eventually goes down into Columbus. It's in the upper part of the East Fork of the White River. But we've only seen 2 live individuals down there in the last 25 years. So that population is very questionable on how it's doing. We have this weird kind of disjunct population, the Salamone River above the reservoir. And it's actually reproducing and doing very well, but it's only in a very small section of the river. And then we have it over in the Tippecanoe River. If you're familiar with your geography of rivers in Indiana, this is the Tippecanoe River, and we haven't really seen live ones in there for a long time. So what we're trying to do with this project is to use some females from the Salamone River population, propagate mussels, and put them back into a spot in the Tippecanoe River to try to get a new population started there. So here we go. This is the life history of a mussel, and specifically a snuff box. So if you kind of start in this bottom right corner, there's your male and female snuff box. They're out in the river. A male releases sperm into the water. The female siphons it in. While filtering water, it fertilizes the eggs inside, and she holds the eggs in her gills. At that point, once they're fertilized, that's the parasitic stage of the life cycle, and those are called Glaucidia. They come in a lot of different shapes and sizes. Unfortunately, this pointer doesn't, it shows up on the wall really good, but it doesn't show up on the screen. But they come in all different shapes and sizes, and a lot of them will be, when they're released by the female, they're kind of open. They kind of look like a Pac-Man. Did you know the video game, a Pac-Man video game? And then they'll clamp shut, and that's how they attach to the gills or the fins of the fish. Mussels have a million different ways to, not a million, but a few different ways to get those Glaucidia to fish. And some of our mussels are very specific in the host of fish that they'll use for reproduction. The best, or one of the best hosts for the snuff box to reproduce is that fish in the upper left-hand corner, which is a log perch. It's a member of the perch and darters family of fishes. It's pretty common throughout Indiana. And the female actually, it's not a great picture here, but the female will gape open her shell a little bit, and there's parts of her mantle that look kind of white that she'll extend out. And it looks like something maybe a fish would be interested in checking out. Maybe it's a food item or something. So that snuff box, or the log perch, will come down there and check it out, and the female snuff box will actually clamp onto the snout of the fish. And then it actually uses some pumping of its gills, and then it explodes out all this Glaucidia into the face of that log perch, and hopefully they stick onto the gills. And then it leaves go, probably being ripped out of the bottom of the stream and laying on the surface somewhere at the bottom of the stream. And then that log perch swims around for about a month with those Glaucidia attached to the gills, and then they are slowly transforming into basically very, very small miniature versions of the adult, and then they fall off that fish wherever the fish is at that point. Yep.
Here's a nitty-gritty question. How does the egg get fertilized and then move to
That's a great question.
gills of the female?
Well, so the eggs are already in the gills. That's kind of where they're produced and held. They're held in very specific sacs in the gills. And then once they're fertilized, that's where they become the Glaucidia, or the parasitic stage. And some mussels actually will put these very elaborate lures outside their shell to attract fish. Others will actually just release these little, they're called conglutinants, or they're little packages of the Glaucidia that look like a little white worm or something that a fish might want to eat. They just expel them from the shell, and they kind of float in the water down to the surface, hoping the fish will eat them. It's a little more random. There's actually even one species of mussel where some of the females of the population actually are very small. They don't get as big as the other females, and they'll actually just lay out on the surface of the water hoping to get eaten whole. Their host is a freshwater drum, and a drum will eat mussels. So they actually give up their life to hope that some of the babies, some of the Glaucidia make it to the gills of fish.
If they get eaten, how do they get out of the digestive tract?
Yeah, so...
They get defecated.
that female is giving up her life, but the Glaucidia, the ones that get digested are done. It just, hopefully some of them make them to the gills. Yeah, and then so wherever that's at, then the fish drops the baby mussels, and then it starts all over again. So what we do is we try to look at two parts of that thing: getting the Glaucidia to a fish, and then where are those mussels going to fall off. Those are the two spots that we're going to try to work at when we do the cage culture and try to do better. And so that's how we can produce. Yep.
So in that picture on the left with the green background and you're looking at the fish's gills, you're looking at those little
White spots.
the black spots.
Those little white spots. Yeah, there's going to be a better picture in a little bit. That same
I was
picture.
looking at the ones where he's lifting up the gill. Whoever it is.
Yeah, that's my finger. Those little, see those little tiny white spots?
The white spots.
Yeah, those are the actual little Glaucidia attached to the gill.
How big are those then? They're like.
Yeah, I mentioned before, like a grain of sand. I mean, probably a little smaller than that, less than a millimeter.
What's the average?
It's a pretty small fish.
Yeah, a log perch is only, about this. We're talking about a fish about this big. They won't be as big in a picture in a minute here. I'll show you. So here's what we do. It's a 3-day process, and it's crazy 3 days. So we have cages. They have these bottoms. We put substrate in those bottoms, usually a little bit of sand, gravel mix. It's about 1.5 inches, 2 inches thick on the bottom of that. That's about a 2-foot by 3-foot base. You can see the long legs. So we're going to push that into the bottom of where we're going to keep it. And then we have a cage top that goes on top of it, and that's where we hold the fish after we infect them with the Glaucidia. So the idea behind this is we hold the fish in that cage, for that month or so time that those Glaucidia are transforming. And then when they fall off, we catch them in the bottom of that cage, in that trough. So we set them out, and we actually, for this project, we put them in Lake Schaefer. The upper part of Lake Schaefer, we have a spot we put them. We sink those to the bottom, we put the tops on, and that's where we're going to put the fish once we get them infected. So that was day one, we just get them right. Day two, we got to find females. This may be not just one day, sometimes this takes a few more days. So we go to the Salamone River, we look in the bottom of the river using view buckets or snorkel masks or feeling with our hands. And this, sometimes we find them this easy where there's a female like that, sometimes they're like that. Right in the middle, that's a mussel, gaped open a little bit, that's probably a female. And so normally from any year, we usually try to get between 4 to 8 females to use every year for this. And we never use the same ones twice, so we'll mark them, and when we put them back in the Salamone River, if we happen to find one we used in the past, we don't use it again. So that's day two, we find the females. We put them in maybe a mesh bag, we hold them in the river for the next day. So we already got the cages in Lake Schaefer, we have some females ready to go. So on day 3 in the morning, we start off and we have to catch log perch. So this is actually, I don't know if anyone's ever been below Norway Dam on the Tippecanoe River up in Monticello, but this is that area. This is where we go to catch our log perch. They're not that big. That's just to show you, again, they're only about this big, right? This is a very small scale project, but we try to catch about 100 log perch. Sometimes it takes us 15 minutes, sometimes it takes us 2 hours, depends what the river looks like, how high it is, depending if the fish want to cooperate and get caught that day. We use seines just like you're seeing there, we just seine them up.
How long does it take you to find the mussels?
Yeah, I mean, if it's perfect conditions, so this is the time of year, it's usually like April, middle of April, May. Sometimes we have a lot of high water that time of year.
Sure.
If there's high water, it's very difficult. There's no visibility. We do it sometimes with no visibility, and we literally get down on our hands and knees and we just feel around the substrate trying to find females.
So is it like hours to find the...
It could be hours.
Okay.
Now this is the snuff box, right?
Just we're talking about the snuff box. Yeah, and maybe that didn't give a good idea how big they are, but you know, a female snuff box is about that big. And so we have the cages already in the lake. We have the mussels ready to go to Salamone River. We've collected the log perch. We put them in a cooler. We drive over to the Salamone River where we have the mussels in a bag, and we set up a lab along the side of the river. And this is exactly what we're looking for. So we gape open that female a little bit. And see the white tips, that really bright white inside there? That's the tips of the gills, and those gills are filled up with Glaucidia. And that's kind of what you want to see on a really nice female. So we use a syringe, and we flush parts of those gills with some water into small buckets. I eat a lot of ice cream at my house. That's why I have so many Meyer buckets of ice cream. But they work good. They were not a partner in this project. And so we flush the gills. We collect that Glaucidia just in a bucket like that. And this is what it looks like when you flush the gills. So there's just thousands of these Glaucidia that come out of the gills. And what you're seeing on these, I know it's kind of hard to see, and I kind of blew up this little part right here. But each one of these is, like I mentioned, they're kind of open like that. But when they get on that gill, they clamp shut. And so then we take those and we mix them in these buckets. We put about 10 fish in a bucket. We dump some of that slurry of Glaucidia in there, leave them in there for about 15, 20 minutes. And then when they come out, hopefully we see log perch like that, okay, that have the Glaucidia attached. Then we drive them back from the Salamone River. And this is a long day. And then we dump them in our cages. And there's our log perch in a cage. And then we stick those cages back in the lake. And there they sit for about a month or so with the log perch in them. Because that's about how long it'll take, depending on water temperatures and how things are going. And then those babies are falling off into the bottom there, hopefully. And so then this is kind of what it would look like after maybe a year and a half. So I'm going to show you kind of the progression. So we want to get them to a size that's big enough that we can put some tags on them, so when we put them into the reintroduction site that we can monitor them over time. So that's kind of what like one would look like after being out for probably a year and a half. There it is with the top off. It's kind of interesting, but you're going to notice in the next slide. So you see all those shells in there? That's all Asian clams. So when we put that substrate in there, there's no Asian clams in it. A year and a half later, we pull that cage out. That's what it looks like with all these Asian clams they've gotten in our face. It's just because they're in the system and they get there. But what you can see in this picture also, you see that right here, right there, there, there. Those are the snuff box that have been dropped off and produced in the cage. So we dig through all that substrate that we put in there and we pull out mussels. So this is what they would look like, and look on the bottom there. So we placed the cages with the log perch in on April 20th, and this one was pulled only about 6 months later, a little less than 6 months. And this is actually the size they will grow to in that size, from falling off the fish, landing in our cage, and growing for about 6 months. 6 to 12 millimeters, so not real big, as you can see. They're not big enough to tag or anything. So what we usually do is we don't pull them after just that 6 months, leave them in for another year. So these are the same mussels, but another year later. So this is in August, at the end of August in 2017, 492 days. And here you can see most of them get more than double in size. And you can see on these mussels are just like a tree. Can you see like this little line right there? Right there, right there. So that's about how big they were at the age 1. And then they get that winter slowdown of growth. So they put a ring down just like a tree would. And you can see how much they've grown in that second year. It's quite a variation though in growth. Sometimes some grow a lot bigger than others. That's the same mussels from the last picture after about a year and a half. And then here's what they would look like after 2.5 years. They get all the way up to about 34 to 42 millimeters. Now they're big enough for us to do something with, where we can actually tag them and then monitor them over time.
How do you attach the tag?
I'm going to show you that next. So some of the mussels we just put a small number tag on with just a little drop of super glue. And then other ones we actually put a pit tag on. Do you know what, anyone in here know what a pit tag is?
Passive integration.
Transponder, yes. If you have a pet, your pet probably has one in its neck or somewhere where if you scanned it with a scanner, it would kick out a number. We use the exact same thing. So we use a little bit of epoxy, and we epoxy a pit tag onto the shell. So then we put in the substrate of where we're putting them back in. We can come back anytime later, and we have underwater readers that we can scan over there and try to find where our mussels are at. That way we can then dig them back up, see if they've lived. We can see if they've grown. We can see if they've moved, if we can find them. Sometimes they move and we can't find them. And then this is where we put them. So this is the Tiffany River in White County. This is our reintroduction / augmentation site. And to date, we've put close to about 500 or so snuff boxes we've produced with this method so far at this site. And survival has been awesome. We've gone back. So we actually lay out a grid. So we have actually a couple steel stakes in the bottom that we can run a line on. And we put them out right along that grid so we know exactly where we put the mussels to start with. Then we go back and that's where we start to scan for our pit tag ones, one of our pit tags. And interestingly enough, so again, these are sexually dimorphic. So we put all the males upstream of the females, hoping that would help. The males have stayed right where we put them. Literally, they haven't moved hardly at all. Females are very hard to find. A lot have moved. But think about, we're taking this as a good sign because think about how they reproduce. They actually grab a fish right by the snout, and then they probably get ripped up out of the substrate, and then who knows where they end up getting back in. So we've actually found a lot that have moved 10, 20 meters from where we put them. And a lot is interesting at this site have moved kind of down and over to the one side. So we're hoping that that means that there's been some reproduction actually happening at our reintroduction site. Yep.
Have you attempted to try to reduce the Asian clams that are in the cages?
Yeah, so we've tried some different size cloth on the outside of the cages. We just did that last year, so we don't have any results yet to see if that has helped. One other problem we have is that when that baby mussel falls off the log perch, it doesn't always land in the cage. So there's actually a lot of snuff box that are actually around where we have our cages in the substrate of Lake Schaefer, which isn't a bad thing because where we're putting these isn't that far upstream, but we like to catch as many as we can. So that's also why we're trying the smaller screening on the outside of the cages, hoping they'll keep more inside. So again, you remember the first one? We're kind of disrupting natural things by us getting the locatium and putting them on the fish, and then us hopefully catching the mussels coming off the fish. All right, so that's one project. What time is it? Okay.
What's the average lifespan of that mussel?
A snuff box is probably 20 plus years. They could easily, that's a younger one though. They don't tend to live quite as long as some. I mean, we have other mussels that can live 70, 80 years.
So they live 20 years, they're harvested, or they're just left in the river? And
These are not harvested, no. especially because they're fairly endangered. But we don't have any harvest of anything in Indiana right now. So these are just, we're trying to get populations back. Number one, so they can come off the federal list, so they're not endangered anymore. And then hopefully they're back in, and the diversity that we used to have will be back in these rivers, and they'll be doing their job that they're supposed to be doing in the rivers of filtering and doing other stuff. So we're hoping if we can get enough populations started that some of these will come off our lists. That's kind of the goal of some of the recovery plans for these species.
So once you get to a certain point, you use mathematical modeling to predict the future a little bit.
Probably, yeah. Yeah, we don't have very many out there. And so this project has been small so far. We got like maybe 500 we put back in just one spot. We'd like to see these like, you know, throughout the river. So.
This is probably a silly question, but are mussels grown commercially anywhere anywhere in the world? Do people actually intentionally grow them for some kind of commercial use?
I don't know for commercial uses. There's a lot of propagation facilities across the U.S. now,
Right.
And most of them are doing stuff like this, recovery of species.
So pretty much they're not used commercially.
There are still a few states in the United States where you can harvest a few in very limited areas. And I don't know the answer to this question on what has happened to the Asian cultured pearl industry, but I feel like they found another nucleus that will work.
Okay.
But I don't quote me on that because I'm not 100% sure.
Sure.
But once a lot of our states stopped doing it, they had to come up with probably something else to make cultured pearls. All right, so I'm going to go kind of quickly through the next one. This is a project we just started this past year. And so besides doing the cage culture, you can also just do captive rearing. And so we're doing a project in the upper West Fork White River with 2 species of mussel, kidney shell and wave-eared lamp mussel. These are 2 species that are on our special concern list right now. And these are 2 that we'd like to see the populations doing better up in that upper White River. So to start off with year 1, we actually used a very common species. This is a plain pocketbook. It's found throughout Indiana. You may have run into this in a stream if you've been out before. It's pretty easy species to work with. It uses a wide range of hosts for reproduction. It's not like very specific to one species. And it grows in a lot of different environments. It's a pretty easy species to work with. So in captive propagation, it's very similar. So we went out, we collected plain pocketbook females that had a glaucidia in their gills. We extract that the exact same way we did with the snuff box, and we infect, in this case, largemouth bass. But instead of putting them out in cages and waiting for the mussels to fall off there, we actually keep them in lab situation, and we wait for them to fall off in tanks. So we'd set up several tanks, we'd infect fish, put them into the tanks, and then there's like a daily siphoning out of the bottom of the tanks through very fine mesh, and we pull out all the little babies that have fallen off. And then we put them into kind of, see the smaller tanks on the up on top of that aquarium? We put them in kind of grow out tanks till we can get them to about maybe a millimeter size. We're talking very small mussels here. And we feed them in the lab using a kind of a commercial diet of different kinds of algae and some other stuff that we feed them. So it's kind of this was year 1. And we were doing this in partnership with the Muncie Bureau of Water Quality, and this is actually in their lab at the wastewater treatment facility. They're right along the banks of the White River. So we trying to do it, try to learn the methods, trying to get some success with a very common species, and then moving on to the kidney shell and wave-eared lamp mussel. So once we got them to about a millimeter length, we then actually tried 2 different ways to kind of grow them out and get them bigger. One was this stream-side setup. So we actually, that levee behind there is actually the White River's right on the other side. We had a pump, and we pumped water from the river into these tanks, and we held those small mussels in some trays in there, using that as kind of a way to feed them and get them to grow. Up there in the upper left is kind of what they look like by the end of the summer. That's very small mesh, you can see, not very big, but we did get some out of it, so it actually worked. But a bunch of other ones, we took and we put them in cages and we put them in Prairie Creek Reservoir. And the upper left is what they look like when they came out at the end of the summer. So about 10 times bigger, maybe even bigger than that. Obviously the reservoir had food that the mussels really liked, and the White River did not have as much of that food. So we learned a lot in that first year. We will probably, I don't want to say we're going to ditch the streamside setup. We might try it another year or 2, or maybe try to supplement with some other food. But literally, you take that 1 millimeter mussels, literally like in a beaker, and you set up a cage in Prairie Creek and you go and you just dump them in there. And then you come back 3 months later and they look like that. You know, you're dumping something in there that you can't even see basically almost. And then you come back and you take them out of the cages and you get hundreds of those. Really nice size.
Were you surprised when you saw them?
I was very happily surprised. I might have screamed and yelled when I opened up the cage and there they were. But it was very exciting. It was our first time in Indiana to actually raise them here ourselves. There's always, like I said, there's always some other facilities that are doing stuff around. And we try to do it on the cheap. Like, I don't want to make it sound like Indiana's cheap, but we try to do it very much on the cheap. And we did this very cheaply. And what the idea of is, if we could do it, you know, man, we could expand. We could get some schools, you know, maybe working on it. We could get some other places that for very minimal cost to get a few things, supplies, we could actually have people kind of raising mussels for us. So that's why we did it that way. I mean, there's other facilities that are very high tech, and they probably have a little more success than we did. But we had success doing it very much on the cheap. Okay, so that's just another, we're just getting that started, and we're hoping to up that this year and try some stuff. We have some hosts. So the one species we want to work with is kidney shell, and it uses the fantail darter as a host, which is another type of darter, so about this big. We've already went out this fall and collected some. We have them in the lab, ready to go this spring. Now our next thing will be is just to find some gravid kidney shell that we can use to get the Glaucidia to infect them with. But so we're going to be moving forward with that, working with Muncie Bureau water quality going forward. All right, so one more project I want to talk about, and this one is very straightforward. This is literally finding a place that has a lot of mussels and taking some adults and moving to a place that doesn't have any mussels. So it sounds very simple, right? Okay, well, here we go. So there was a bridge project going on the Allegheny River in Pennsylvania. And it just so happens that they're going to tear this down. Well, they already did now, but they were going to tear it down and replace it with a brand new bridge. It just so happened underneath that bridge there's 5 federally endangered mussels living. So of course they had to do something with it. And these are some of the estimated abundance of these federally listed species in the area that would be impacted by the project. And you can see large numbers of a couple species, especially northern riffleshell and clubshell. So they're like, well, in most of these types of projects, they'd go in, find all those mussels they could, and just move them upstream a little bit in the same river. But they decided, well, you know, there's already a lot of these mussels. This is the thickest amount of clubshell, northern riffleshell anywhere in the world was in the Allegheny River through this stretch. So instead of just moving them somewhere that already has a lot, why don't we do something to try to help recover these species throughout their range? So they decided instead of just moving that thing that they would give them to some states to try to reintroduce into some places where they used to be and they weren't anymore. So we jumped in on that project. To give you an idea, here's northern riffleshell. This is the last 30 years worth of surveying in the state. And you can see there's no red or orange dots. Remember the red and orange dots where I had seen live ones or where we've collected live or fresh dead. We have not seen this thing live anywhere in the state in the last 30 years. So we decided to choose an area on the Tippecanoe River where we have other good mussel populations to do an augmentation. We're calling it an augmentation. It was really a reintroduction of northern riffleshell into the Tippecanoe River. And then with clubshell, we do have a couple populations. The upper Tippecanoe River, we see all those red dots together. There's still a reproducing population in there. But you can see how widespread this species used to be at one point in Indiana. It was basically in almost every river and stream throughout the central part of the state. So we decided with this one that we would reintroduce a population from the ones we could get from Allegheny River into the Eel River. So the Eel River is kind of up. It comes in by Logansport into the Wabash River. It's the next big trib east of the Tippecanoe River. And so that's where we targeted to do a reintroduction with clubshell. This just gives you an idea. That star is where that bridge is at. And this is where mussels from that bridge went. All these different states into all these different rivers and streams to try to get these populations kind of restarted or reinvigorated. So we first went out and we set up some sites, plots, so we laid out a grid line and we put markers in so we could put these very precisely in spots so we could monitor them very easily. We did a survey of what native mussels were there before we got the other ones. The spots in the eel and the tippy both were very diverse with some native mussels. These are 2 of our best mussel streams in the state. And then they went out and they started picking up all the live mussels at the bridge project, and they all got tagged. You can see the little number tag, and then you see that little bit of epoxy below it. That's where a pit tag is placed in it. And then they sent them to me in a cooler overnight. And we took them This was back in 2015. We took them and we placed them in northern Riffleshell in the Tippecanoe River and Clubshell in the Eel Rivers I mentioned. So we hand placed them right where we knew where they'd go, and then we monitored them for a little bit. We only got in that initial thing a small amount just to see how they would do. It's kind of a pilot study. And so we got 150 in October, in September, and then we monitor them into June of the next year. Now you can see in this one, the northern Riffleshell, that's one of our ones we got that was buried in, looking really good. 11 of 15 that we checked in June were found to be alive. So we did lose some mussels in this transfer. Clubshell, all were alive, right where we put them. They all look great. And so because of that, we did as well, if not better than any of the other states. We got 3,000 clubshell and 3,000 northern Riffleshell the next year, or later that same year. And so we went back out, and as you can imagine, putting that many mussels in the river, along these grid lines, we got a lot of help from some of our partners, TNC, U.S. Fish and Wildlife Service, some of our other DNR employees, and Manchester University helped us out on the one river. Got a bunch of students out there. And we put all these mussels out, and that was back in 2016, so we've been monitoring them since. And again, we can monitor the pit tagged ones because we can use underwater readers and find where they're at. And then we can dig some up, see if they're alive, see if they've grown, and then try to see if they've moved at all. So here's kind of what we've seen so far. With the northern Riffleshell on the Tippecanoe River, you can see the numbers down there that we originally placed. So the pit tag's 442. Unfortunately, we've already seen close to 200 of those that we know are dead, and another 87 that we've not ever been able to relocate. And the glitter ones, so some of them, let me go back. Up in the upper left, if we didn't put an actual number tag or a pit tag on, we at least put a little bit of super glue with some glitter on them, so we knew it was a mussel we had placed in the river. So once we're hoping reproduction happens, we want to be able to know that it's not a mussel that we just found that was already in there, that we had placed. So they either had glitter or that, so they're all marked. So unfortunately, northern Riffleshell, and this was actually seen throughout all the states. Survival was okay, but it wasn't, I wouldn't say it was great in any length of the thing. But it was very similar. And the good thing is we do still have some that have lived up to over 5 years since we put them out. So that is actually positive. The one thing we haven't done yet is we try to find juveniles, because ideally we want to see these things reproduce. If they never reproduce, then it's kind of pointless. We're just putting a bunch of mussels back in the river, okay, but we want to see them reproduce and then start to expand their range. And so that's kind of on the horizon, is to go out and actually try to find babies, which is a very difficult thing to do, but we're going to try. Clubshell, on the other hand, the numbers there that we put out through 2020, September 2020, hardly none we have found dead. They've almost all survived, and they've almost all stayed right where we put them. They've been awesome. They've grown. We're really expecting to see some reproduction. Certainly, they've probably reproduced by now. So again, next year we're going to go out. It's kind of hard to know where to look, so we put them all in this one 8 meter by 20 meter area of the river. But once they get on that fish and they ride around on a fish for a month, who knows where they're going to go and fall off. So maybe some falling off in that same area. Maybe they've fallen off a mile below, a mile up. We've actually considered doing some eDNA studies. So actually taking some water samples just above where we placed all these mussels. And we know there aren't any above it naturally. And then if we get hits through DNA through the water, we might know that some actually have fallen off upstream. That might be something we try. Otherwise, we're just going to have to get out and do some digging around and hopefully see if we can find some babies. That was through 2020. I am going to show you one thing that happened at one of our... So great survival, hardly any dead. It just so happened in September of 2021, we came to one of our sites and we saw this. I don't know if you've ever been out in a river and seen what's known as a midden. So it's a pile of shells. You can see it there in the middle. All in a nice, neat little pile, huh? Muskrat. So a muskrat got into our site, unfortunately. And here's its den, or one of the dens. And you can see by its den, there's a pile of shells right above it there. Well, that's how many it got of our mussels at that one site. Well, and it wasn't just one muskrat, obviously, it was a family of muskrats. But we went through every midden we could find on both sides of the river. Over 150 of our about 1,000 mussels at that site got eaten by that muskrat family within a couple weeks. So unfortunately that happens. Again, you never know what's going to happen, right? But we're still very hopeful that they've been out there long enough now that they're reproducing on their own, and we'll see some reproduction. Yep.
So how close do they have to be to successfully reproduce?
Yeah, that's a great question. So Yeah, you know, you think about a male releases sperm into the water. You would think that has to be a fairly close proximity for a female. The sperm can last in the water for a little time, so you could probably make it. You know, I don't want to hypothesize on how far down a river the sperm can make it until it gets a female, but it's probably not, doesn't have to be just this close. You know, it can survive probably in the water for a little while. But yeah, you would think that you'd have to have them in close enough proximity in order for reproduction ever to happen. And that's the problem. Some of these populations have gotten so small, the males and females just aren't close enough to each other to actually
It's not
interact.
gonna do any good if it just swims a mile upstream
Right.
and we drop off up there.
Does the temperature make a difference?
Yes. So that's probably what triggers some of the mussels to release Glaucidia, like in those conglutinates or to display. Mussels are very different to different species. So some mussels are what are known as long-term brooders. So they actually get Glaucidia in the fall, and then they hold them all winter and they release in the spring. Other ones in the beginning of the summer get Glaucidia and they release them within that same summer. Those are short-term brooders. Different mussels have different strategies for how to reproduce. But that would have a lot to do with when they release, have a lot to do with how fast mussels can grow. Definitely temperature will play a key role in both of that. And so actually that question you just asked kind of goes with the last two things. I don't really have good examples of the last two. One would be is that you get female mussels, you infect fish, and then you just release fish back into a stream and let them do whatever they do. So you're taking out at least the one step of the natural reproduction by getting Glaucidia on fish. We have not done that yet, but it might be something we'll do down the road. And that one is just hard to monitor because you're just releasing these fish and then you don't know where, you know, the mussels hopefully will fall off somewhere and do well, but you have no way to know. And then the other one's called aggregating. It's just the one where you do get a population of a species that's really small. You go into a stretch of river and you try to find everyone you can, and then you put them all kind of in one area together. And that's just hoping that you will help them be able to reproduce. So that's called aggregating. We have not done that yet, but that's something we may do shortly with fan shell, which is down the East Fork of the White River in Martin County area, and it's something that's very rare there, and we may go out and try to find someone and get them close together. Yep.
Is the Department of Commerce interested in what you're doing?
That's a great question. I don't think so, no. I don't think they care.
Well, I heard today someone was saying in Kentucky, they're trying, and it's my understanding as well, that both Kentucky and Illinois commercially fish the Asian carp.
Yeah, and we're pushing the same thing. So we actually just hired Asian carp biologist. That's all they're doing is working with Asian carp, and they have 2 assistants. Just got hired just this summer. And this next spring, they're actually looking to hire some commercial fishermen to come into some of our rivers and actually target Asian carp to take them out.
And is there a future in our lifetime? I mean, you know, the mussel population seems so depleted.
It's a big task, let's put it that way.
So,
and it takes time, yeah, for sure.
Yeah.
And then a muskrat comes along and eats most of it, unfortunately. So those are the last 2. And I'm going to do one little plug here. So all the stuff I do, when you do your state, does anyone pay state taxes in here? Remember how there used to be that little eagle logo on there? We could donate some of your refund if you happen to get a refund to the non-game fund. Well, they did away with that a few years ago. And when they did that, now you have to go through another form. There's some competing groups trying to get money from the refunds. And our non-game fund donations was like this, and then it went like this when that happened a few years back. And unfortunately, that's what we use to match the federal money we get is that non-game fund. So all the work I do and our non-game mammologist, our non-game bird biologist, our herpetologist, we need that non-game fund. So I got to put a little plug in there. So if you're doing your taxes, you got to do the extra steps to actually donate some of your refund to our fund, which we use, and then we use, like I said, use that as a match. And that's my contact information. I'd be glad to talk to anyone if you have any questions, my email address or my phone number. And I'll put that back up when we're done. And then if you have any questions, I'd be glad to try to help answer those. I mean, we asked a lot, which is good throughout it.
For those that are known, how many of the host fish species, the darters, what's the status of their population?
Yeah, that's a good question. So we know the hosts for a lot of our mussels, and it's just lab experiments. That's basically the way we figured it out. People get a whole bunch of different species of fish. They find a gravid female mussel. They infect all those different fish, keep them in different tanks, and then they just watch for stuff to fall off and see if it's viable. And there's a couple universities that they do it all the time, and they use students to do it. And so we've learned a lot about what hosts work for our fish. Some are very generalist, they'll use about anything. Some are very specific, like there's one here in the Wabash River. It's the fragile paper shell, I might have mentioned it earlier. The freshwater drum is the only host for that species. So you can imagine that their distribution overlaps pretty well. Fortunately for freshwater drum, populations are great. We have them all over the place, and you can pretty much find fragile paper shell about anywhere you can find a drum. For some of them though, yeah, it can become a very big issue if the host populations aren't doing well. There's one very good example, it's called the elephant ear, and its host, believe it or not, is a skipjack herring, which I don't know how many people in here would even know what that is. But it's similar to a gizzard shad. It's in that same family, the herring family in Indiana. It's in all of our big rivers in the state. But in the 1920s, they put a dam in on the East Fork of the White River, Williams Dam. You might know of it. It's the town of Williams. And at that point, there were elephant ear populations upstream and downstream of that dam. And at that point, those skipjack herring could no longer really get up. And any populations that were above either came down and went over or just kind of went away. So we don't have skipjack herring above that dam anymore. Well, the minute that happened, all those elephant ear that were above Williams Dam, they're just living out their lives. They're never going to reproduce again because their hosts can't get there anymore. Weirdly enough, they use the skipjack herring. But yeah, so we have very good examples of that in the state where, you know, the host can no longer get there. It's a big problem. And then think about the Ohio River, how much that changed when they put all those impoundments in the Ohio River. Think about like species that maybe used to live in some of those big, huge riffles that were in the Ohio River. Now you don't have that, so some of those mussels took hits too because their fish hosts just weren't around anymore, didn't do any good. That's the thing about fish, you know, fish can too swim back in. We talked about this earlier. A big event comes through, like the White River fish kill that happened like in 2000. You go back there now, you never know there was a White River fish kill. The fish populations are as good as they ever were in there. But if that happened for mussels, like if something came through and wiped out all the mussels in the west, luckily that did not affect our mussels in that river. But if it did, I mean, the years it would take for them to get back in there, and if they ever could get back in on their own, and if there's a source population close enough that maybe every now and then a fish would swim upstream and drop off a few baby mussels, I mean, it's crazy that they would. That's why a lot of these streams we have are in problems, because they'll never get their mussels back without some help.
Do the water moccasins ever bother you?
The water moccasins. You mean just being out in rivers and streams?
Yeah, when you're out doing your mussel thing, I mean, do they come around and bother you, or just...
I see water snakes every single day I'm out, but I've not seen a water moccasin.
I mean, do snakes kind of steer clear of you when you're doing...
No, not at all. I've had them swim right between my legs, literally. Yeah, no, we see snakes every time we're out. They're always out and about.
So have you ever been bitten?
No, never been bitten.
Yeah, they are poisonous though.
Well, a water moccasin would be, but we maybe have a very, very small population in one swamp in southern Indiana that used to be there. But what we have is banded water snakes that are literally in every stream in the state, and they're abundant. But yeah, yep.
So how is the mussel population specifically like in this area? Like, is anything that we're doing like with the Wakashiki helping it, or, you know, is the Wabash like beyond hope?
I don't want to say it's beyond hope, but I did, and I don't know if you've been to the exhibit yet. I think there's some actual numbers there, but I did bring this. So like if you looked historically at the main stem Wabash River, I'm not talking about tributaries into it, but there were probably about 66 species there that were living in the Wabash River. Maybe not all 66 right through this stretch of the Wabash, but the whole length of the Wabash. 18 are pretty much gone. They're not living anywhere in the Wabash. 18 other ones might be reproducing in a tributary to the Wabash, but they're not reproducing anymore in the main stem Wabash River. So that leaves about 30 species that you could now go into the Wabash River and say are still reproducing in the river somewhere in the length of it in Indiana. So that's about 45% of what used to be there. But the ones that are there, there's a handful that aren't doing so well, but the other ones are actually doing okay. And that's because they're all ones that can live in about any type of habitat. And they almost all have drum or catfish as hosts for reproduction. As you know, there's a lot of drum and catfish in the Wabash River. So those are the ones that are doing well now. But I don't know what the habitat or what the Wabash looked like 200 years ago, but I think it looks a lot different than probably what it looks like now. It's a very mobile stream, like the substrate is very shifty. There's a lot of sand. Mussels need like stable substrate. They need something they can bury down in and feel good and not have to worry about getting blown out and moved around. And I feel like the Wabash is not that in a lot of places, especially this lower section.
How deep down do they go with the mussels?
Some will bury to the point, like, say there's a mussel the size of your fist. Some will bury in so you can only see just the very tip top of where they're siphoning water. You would never know they were there if you weren't looking close. Other ones stick out a lot more than that. But they'll bury in pretty deep if they can.
Completely bury themselves.
Not normally. There are some that will do that. If it's really like kind of a gravel cobble mix that's real clean, they will bury, some species will bury pretty deep in that where you wouldn't even know they were there at all. But most of them, usually you can see that very tip of their shell or where they're siphoning water is usually exposed.
What's happened to the market that went away in the 90s?
Yeah, that's why I mentioned that earlier, and I don't have good information on what the Asian markets are doing for the cultured pearl industry anymore, but they obviously are still making cultured pearls. So I assume that they figured out some other way to make cultured pearls without using our mussel shells as the nucleus. It was just the best nucleus, so that's why they were using it. They would take that, those thick-shelled species, and they would cut them down into little cubes. Then they'd grind that down into what would look like a pearl, and then they actually surgically implant that into a pearl oyster, leave it in there for about a year or two, and then harvest that pearl back out and sell it as a cultured pearl. And that's what they were using our shells for, obviously because it was the best nucleus. I don't know what they're using now, but they're still probably getting some from a few states that are selling it, but does anyone know if the cultured pearl industry is still a thing? I assume you can still buy cultured pearls, right?
I know they're doing a lot of culturing of different kinds of oysters, like in Mexico on the Pacific coast, they've got the fan oysters. I can't remember what they're called, but I know there's a big culture facility there for that. And of course, commercial oysters are cultured. But
right.
You wonder if oyster shells can be used.
Yeah, I don't know. I don't know why they wouldn't use that to begin with. Like, why they go through the whole process of having to come to the Midwest of the United States to get the nucleus, Yep.
One of the other talks spoke a little to that.
Okay.
Because of the high concentrations of calcium in this specific area.
That makes a lot of sense.
Because of all the tributaries, and it has to go through a lot of the limestone, the marble, the dolomite, and that is why we have some really strong calcite-rich mussels.
Yeah, so that makes a lot of sense.
The limestone.
Yeah.
So would you use a scuba mask with an air tube when you're doing your work, or what would you do?
It depends. A lot of our surveys we do would be done with diving. Actually having dive tanks on the whole thing. Some of those, like diving in the Ohio River and surveying mussels in 30 feet of water, there's no visibility. It's all by touch. You're just feeling around and digging stuff up and then putting it in bags to the surface so you can identify it. Some of our rivers and streams in Indiana, though, are actually crystal clear in the summer, where you don't really need much more than just a snorkel mask, or you can just literally see through the water to find mussels that are buried in. We do have these 5-gallon buckets with the bottom cut out that have like a plexiglass on the bottom you can use to view, so you push that down in the water, it breaks that surface tension. We do some streams where literally it's all feel. Even though it's shallow, you can never see, like there's no visibility. It wouldn't even help if you had a mask on, because you still can't see the bottom.
The headlight.
Never used a headlight to look for mussels, no.
Might help.
No. Yeah, well, I take it back. Sometimes in the Ohio River, some light helps. But I've been
With different colors, you can put it on, and you can actually see better with the red.
Yeah, that might be true.
When you go out and you're putting the mussels in the river to relocate them or move them, whatever, do you have to like dig them in, or do you just scatter them? Or
Yeah, that's a good question. So, and you've probably been out, like even on the Wabash River, like in the summer when it starts going down, you'll see mussels with trails. You see these grooves in the sand where something's moving around, that's a mussel. Fall to one end and dig it up, you'll find a mussel that's trying to get back in the water. Or sometimes you find them just laying out exposed. And sometimes people put them in upside down. So usually we tell people there, just lay them in the bottom and they'll find their own way and dig themselves in. But for the stuff we were doing with those, we actually normally would kind of dig a little bit and just push it in a little bit. And you want to make sure you don't put it in upside down. So there is a right way. There's a right way and a wrong way to put a mussel in.
Like planting garden.
Yeah, like planting a tulip bulb, right? You don't want to plant it upside down.
Green side up.
Yeah. Well, for mussels, if you find one or one that's been pulled out of the bottom, just look where the algae's growing, because the algae's growing in this part that's exposed. And usually the rest of the mussel shell is clean because it's been buried down in the substrate. And so that helps you know which side was out. For the ones we do, though, you have to know which one.
How do you know on that one?
Yeah. So that's a mussel that's completely buried in. It's got algae growing all over its shell. So actually, though, if you pulled that out, all that algae would be clumped up right on the top. That's the surface, the spot that should be out, and the whole rest of the mussel would be perfectly clean. I think that's a round pigtail, actually. Yeah.
When you're in the Wabash River, like how would you get off of the water, the pollutants or whatever's in the river? I mean, do you like have some kind of special method to clean up after dying?
I don't do anything really very specific. Yeah, so we do work all over the state of Indiana doing different surveys. Like those maps that showed all the dots, that's all because we went and looked everywhere for these things over the last 25 years. We've been in some places that were usually we were waders in some spots. Sometimes we just wet wade and go in mud and your hands come out a different color by the end of the day. But I don't know. Yeah, there's probably a very good way to clean off from that, but we don't know. I don't normally do it.
Is there a water temperature where it's too cold where you just absolutely would not go in?
Yeah, well, and for the mussels too. So we try not to do surveys when water temperatures are below 50 degrees. Because then the mussels, like you mentioned, like you have to push them back in or can you just lay them out? If it's warmer, you can probably just lay them out and they'll find their own way to get back in the substrate if they can. But when it gets that cold, sometimes we worry a little bit about they won't. Because, they're just going to be a little slower and not be moving as fast either. So we don't normally do surveys when it's below 50 degrees. Not that I'd want to be wet wading at them.
What can you tell me about who and how named these mussels? And is it old folklore or?
Yeah, so that's a great question. So they all have, a genus species name, right? But the common names, yes, the common names are just common names that someone gave them a long time ago. And local people around here might call some of these by different names, a common name. In fact, there were some very colorful common names for some of these mussels way back. But like for instance, like these mussels here, like I'll start in the upper left and go across, and then go across. That's a maple leaf. That's a cylindrical paper shell. This one's a purple warty back. And that's because the inside of the shell is purple. So if you open that one up, that's the kidney shell we mentioned before. That's a spike. This is just a pimple back. This is called a creek heel splitter. This is a lilliput. These are not by size. So the lilliput is actually, it only ever gets about that big at maximum size. This is a pink heel splitter. Again, a heel splitter. I mean, you can think about how some of these names got, right? It's either the shape, what they look like, or someone stepped on it and they hurt their heel. And they're like, that's a heel splitter. This one actually used to be called a squawfoot, but they changed the name because squaw wasn't that great. So it's called a creeper now, which I don't like the name creeper either. That seems kind of weird. This is the fragile paper shell we mentioned before. This is called a rainbow, pistol grip, deer toe. This one's called a washboard. So it's got all these ridges and like bumps on it. You can think of like an old washboard. And this one's called a three ridge because it literally has 3 ridges.
So, but they actually have
right. So like that.
magical name.
Yes, exactly. So like that 3 ridge, it's actually Emblema plicata is the genus species name.
Okay. Thank you.